Hello, it is Joanne again. This video will be discussing DNA replication. Each new DNA molecule contains one old strand. This is known as semi-conservative replication. There will be an old strand as a template and one new strand as a copy. DNA is very stable because of the high numbers of hydrogens between the bases. In order to separate DNA, if we were in a lab, we would use high boiling temperature. But if you are a cell yourself, you cannot physically heat up to the amount needed to separate those bonds. In order to do that, the cell has its own way and mechanism to do so. There are sequences known as origins of replications, which are known as limited areas of replication. These areas can also be known as replication bubbles, and they are localized regions. In these replication bubbles, there is a replication fork. This is the area of the replication bubbles that are being pried open. They are bidirectional because they move in opposite directions. They move from 5' to 3', and the strands are anti-parallel, hence bidirectional. In this replication fork, there are many enzymes, many proteins, and many components that must be aware and known. There is a leading strand, which is known as the strand that is continuous in replication. The lagging strand has to be replicated 5' to 3', but it isn't open fully, so it must be in discontinuous segments. That is known as the lagging strand. And those strands give off these fragments called Okazaki fragments. In order to keep the replication fork as functional as possible, DNA helicase will spin very fast using ATP to pull these strands apart. The strands may be able to zip closed after DNA helicase, so in order to prevent that from happening, single-strand binding proteins are used. They bind to single strands and keep the strands from reassociating together. Using DNA polymerase, a new phosphodiester bond at the phosphate group of one of the nucleotides is created, and the hydroxyl group with the hydroxyl group of the following nucleotide. This bond allows for the creation of deoxynucleotide triphosphates. Each nucleotide will be in this structure when entering DNA polymerase. DNA polymerase has multiple domains or reaction sites, such as the polymerization domain. This is where DNA is synthesized. And there's also exonuclease domain to fix maybe some mix-ups at the same time and clip DNA when needed. When cleaved, there would be large energy for DNA synthesis. In order to keep DNA polymerase from floating away and being as quick and sufficient as possible, there is a clamp protein. This clamp protein encircles DNA and interacts with polymerase personally to keep it bound. The clamp has a loader. The clamp loader binds to the clamp and uses ATP to open and close the clamp when intertwined with DNA polymerase. The clamp loader may also remove the clamp, and allowing DNA polymerase to be removed. The lagging strand, unlike the leading strand, must do this a numerous amount of times in order to continue to replicate as the replication fork continues to open. In order for DNA polymerase to do its job, it needs an RNA primer. This primer is used in order for DNA polymerase to begin on a double-strand landing pad. It cannot start on a single strand. RNA polymerase, on the other hand, can do the single strand. The leading strand needs only one RNA primer, whereas the lagging strand needs many. And that is because of the continuation of the leading strand and the segmentation of the lagging strand. So each segment needs a new RNA primer. In order to bind these Okazaki fragments together, DNA ligase is used. Once the primers are removed, DNA ligase must fuse together those individual segments. And the repair polymerase will add new DNA where there was once RNA primer. And DNA ligase will join the backbones of these fragments and have one single strand at the end of replication. DNA will add caps for RNA primase binding. These caps are known as telomeres, and they eliminate shrinkage of DNA sequences that are important. So telomerase makes telomeres at chromosome ends, which make a continuous strand in order to properly let DNA polymerase to finish the replication needed at those ends, rather than shortening at the ends. If you look here, you can see the different components I have talked about. You can see DNA polymerase moving down this 5', this is the leading strand, so you can see DNA polymerase moving down the 5' to 3' strand. Where if you see on this side, the 5' to 3' has not been fully opened. You have the 5' over here, and then the 3' at this end. So you can only do so much until it is time to add a new DNA primase or a new primer. A better picture is here. You can see the clamp that I talked about attached to DNA polymerase, and you can see the clamp loader holding that clamp and opening and closing it when needed. Here you can see the leading strand, 5' to 3', and you can see that DNA polymerase is moving down this way. You can also see DNA helicase breaking through and opening those strands. Here you can also see the single-stranded binding proteins that are allowing this strand to stay open, although DNA helicase is already passed. When you look at the lagging strand, you can see that DNA polymerase had to start here. And that is because the 5' end is down here, 5' to 3'. So it must start right here, which is where the primer will be, in order to allow DNA polymerase to replicate this strand. And this will be known as an Okazaki fragment. You can see the primer here of this first strand. This, you can see the telomeres and the telomere replication. So this is a missing DNA on the lagging strand. So the lagging strand just had the primase here at multiple areas. When you remove that primase from that top strand here, you're missing the rest of this replication. So in order to continue this replication, telomerase has a sequence that can be added to the lagging strand in order to allow DNA polymerase to come through and read the rest of the missing strands.